Welcome to this explainer. Okay, today we are looking at absolutely one of the most incredible pivots in the entire history of science. We're talking about humanity's massive leap from just, reading our own genetic instruction manual to actually precisely rewriting it. That's right, we are diving deep into the molecular architecture of life itself. Okay, let's dive into this. Here is our roadmap for today. We're going to start with the code of life, then move into unlocking the double helix, explore CRISPR gene editing, and finally navigate the ethics of enhancement. So part 1, the code of life. This is where biology goes from a purely descriptive science to a strict molecular discipline. For a really long time, biology was basically just about observing physical traits. Back in the 1860s, Gregor Mendel figured out that traits were passed down through discrete units. But here's the crazy part. For decades, nobody actually knew what those units were made of. A lot of scientists actually put their money on complex proteins carrying our heredity. It wasn't until the 1940s and early 50s, thanks to the brilliant work of Oswald Avery and the Hershey-Chase experiments, that the scientific community finally proved that DNA, and not protein, was the true molecule of heredity. And just like that, suddenly life was understood as an information system. Francis Crick came up with what he called the central dogma of molecular biology. Think of it literally like executing code or following an instruction manual. First, DNA is transcribed into messenger RNA. Then, that mRNA travels out to these little cellular factories called ribosomes. And there, it's translated to synthesize the proteins that actually build and run your entire body. It is just this beautifully organized, non-stop flow of biological data. All right, moving on to section 2, unlocking the double helix, the architecture of life. So knowing that DNA carried the code was one thing, right? But how was it actually built? To figure this out in the 1950s, James Watson and Francis Crick started applying physics and chemistry to what was essentially biology's biggest structural mystery. But they desperately needed hard data, and they found it in Rosalind Franklin's incredibly precise X-ray diffraction images over at King's College London. Her famous Photo 51 provided the undeniable, rock-solid visual proof of a highly ordered helical structure. That photo was literally the foundational clue that made the whole puzzle fall into place. By playing around with metal models to test out different arrangements, Watson and Crick officially proposed the double helix structure in 1953. But honestly, the absolute most crucial part of their model is the rungs of that twisted ladder. This is the principle of complementary base pairing. Adenine always pairs with thymine. Cytosine always pairs with guanine, locked tightly together by hydrogen bonds. Now, why does this matter to you? Well, this exact chemical pairing is the fundamental reason why you look like your parents. Because each base has a strict, specific partner. One side of the DNA ladder acts as a perfect template to build the exact opposite side. In their groundbreaking 1953 paper in the journal Nature, Watson and Crick dropped what is arguably the greatest understatement in the history of science. They wrote, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." With just a touch of dry, scientific humor, they quietly announced to the world that they had solved how cells replicate with such high fidelity, ensuring all that complex information passes accurately to the very next generation. Which brings us to section 3, CRISPR, editing the genome, or precision surgery for the 21st century. Okay, major pivot here. We are now completely leaving the era of simply reading the genetic manual, and we're entering the wild new age of actively rewriting it. And what exactly makes this possible? CRISPR. The crazy thing is, its origins actually lie in an adaptive immune system that bacteria use to remember and chop up attacking viruses. Then, in 2012, Jennifer Doudna and Emmanuelle Charpentier brilliantly engineered this natural bacterial defense system into a fully programmable lab tool, CRISPR Cas9. I want you to think of this process like highly precise molecular scissors. It's basically a 3-step process. First, a synthetic guide RNA identifies and locates the exact target sequence on the DNA strand. Next, the Cas9 enzyme steps in and physically cuts the DNA at that specific spot. Finally, as the cell's natural repair mechanisms kick in to try and fix that break, scientists can step in and harness it to either completely disable a faulty gene or even insert a brand new one. And this brilliantly illustrates the sweeping, real-world impact of the technology, because it's so much faster, simpler, and way more cost-effective than anything we've had before. CRISPR is revolutionizing, well,
Everything. We are investigating complex conditions like cancer and neurological disorders. Right now, there are clinical trials exploring treatments for previously incurable single-gene disorders like sickle cell anemia. And it's not just human health. In agriculture, we are actively securing our global food supply by developing pest-resistant crops and engineering plants capable of surviving extreme climate-driven droughts. Biological systems are officially becoming objects of deliberate technological design. But of course, with all this comes Section 4, the ethics of enhancement, navigating the biological frontier.
Now, with profound power comes the absolute necessity for profound responsibility. And right now, a highly contested debate in bioethics revolves around therapy versus enhancement. Therapy is pretty universally understood. It's healing the sick or correcting a genetic defect. But enhancement, that's using biotechnology to push physical or cognitive capabilities way beyond normal human limits, like dramatically extending lifespans or fundamentally altering brain chemistry for better focus. The major societal concern here is resource availability. If only the wealthy can access these tools, we risk creating permanent biological disparities right alongside our economic ones.
To navigate this minefield, bioethics integrates scientific knowledge with human values, relying heavily on 4 core principles to act as our guardrails. First up is respect for autonomy, ensuring individuals actually have agency over their own biological interventions. Then there's beneficence, which is the drive to promote overall well-being. We pair that directly with non-maleficence, the classic medical mandate of avoiding harm. And finally, justice, making sure there is a fair and equitable distribution of these revolutionary resources. These principles are absolutely vital to ensure that technological progress benefits all of humanity responsibly, because without those guardrails, we face unprecedented historical flashpoints. Back in 2018, the scientific world was stunned when researcher He Jiankui announced he had used CRISPR to edit the human germline. Now, germline editing means altering the genetic material in embryos, which means those changes can be permanently inherited by future generations. This was widely, globally condemned. It effectively forced society to confront the immediate, very real dangers of permanently altering the human gene pool without long-term safety data or any sort of widespread ethical consensus. Which essentially leaves us here. For the very first time, humanity is intentionally influencing evolutionary processes that were once entirely beyond our control. We are the active editors of our own biological source code now. As we process this incredible leap from Mendel's pea plants all the way to CRISPR's molecular scissors, it leaves us with a lingering, profound question. As we take direct control of our own evolution, what does it truly mean to be human? Thank you so much for joining me for this explainer. Keep learning and keep asking those big questions.